All right, everyone. There we go. There goes my screen. Okay, hi everyone. Let's talk mitochondria. Now, remember, mitochondria are a little special. We believe that they developed through the idea of endosymbiosis. That means that they were engulfed by a larger cell, and then that cell and the smaller cells created a symbiotic relationship. So the mitochondria were allowed to live inside of that cell. And just as a review, there's lots of evidence for this. First of all, mitochondria have their own DNA. It's circular and naked, just like prokaryotic cells. They also have ribosomes that are the same size as prokaryotic cells, that 70S versus the 80S, which is found outside in the cytoplasm of the cells that they're in. So if this happened, this allowed the mitochondria to develop their structure in order to really efficiently do nothing but cellular respiration. So there's some special structures that mitochondria have that allow them to do their job very, very efficiently. So these are two mitochondria, just in case you didn't figure that out. So I know they're kind of round. They're not all elongated like that little kidney bean shape that we always see them drawn in. So there is a double membrane, and these lines, these are membrane that extend off of the inner membrane and into the matrix of the mitochondria. So you have an outer membrane, an inner membrane, the cristae, and then the matrix. So let's talk about this. So remember, this is the powerhouse of the cell. It makes all of the ATP that cell needs. Remember that cells cannot borrow energy from each other, so each one has to be able to do this. Okay. And they do, if it's inside of the mitochondria, it's always aerobic respiration. Outside the mitochondria, it's anaerobic respiration. Okay. So the outer membrane contains the transport proteins that enable pyruvate to come into the mitochondria. The inner membranes, this is where the electron transport chain is, and this is where the majority of the ATP is made during cellular respiration or oxidative phosphorylation. The cristae, those are the inner membrane folds that extend into the matrix, and their whole purpose is to increase the surface area and volume, therefore increase the amount of space that the electron transport chain can happen in and be able to create more ATP. You have the inner membrane space. These are the small spaces between the membrane, and they have to be small because we need to build up a concentration of hydrogen to push back through ATP synthase in order to make lots of ATP. And then the matrix, that's your central cavity that contains all of the enzymes, and it also contains your ribosomes. And it really does allow for the correct pH for the Krebs cycle. Okay, so here's a simple diagram. Your assignment is to annotate a diagram on a Google drawing. I would really like you to use the book, page 387, to help you with this. This is very simplified. I need to make sure that you understand where everything takes place. This is a very complicated, drawn-out process, but you got this, and we can do this. So hope this helps you understand that, you know, mitochondria are a little special. Chloroplasts are too. We'll get there next week. Anyway, hope this helps, and I will hopefully see you in class.